pour vos attentes. Donc j'invite sans plus tarder Dr Hernan Pinto à nous faire sa présentation.
Désolé, mais mon français c'est très limité, alors je ne peux pas faire la présentation en français. On va continuer avec l'anglais. Okay, guys, so sorry, thank you for staying this long. I know you're tired, I know it's been a long journey. Thank you, Amin, for the invitation. So we're going to talk a little bit about MCT and mainly about exosomes, autologous exosomes, which I know you are really interested in. But first of all, let's take a look at the platform. This is MCT. The two parts of the system is, do I have a pointer? No? Okay, no worries. So the small green thing over there, it's the kit, which is a consumable. It's what we use in every session. And the other white thing is the machine. We call it unit, and it's what sends the energy to the sample. So we started this journey a long time ago. I know because we changed laptops and all this, you see some letters are not very well arranged, so sorry about that. We started a long time ago seeing some papers that talked about increasing the amount of growth factors when we use light, or increasing the amount of growth factors when we use temperature. This is all very well documented. So we started digging on these facts, the fact of how can we affect cells and platelets with temperatures and with lights. Just that, natural things, no chemical additives, no whatsoever. So we reached some studies that talked about physiological properties of PRP to different pH, etc., etc. So it was clear that temperature and light would affect the samples, but we just needed a lot more to understand. Thank you very much, Amin. You're very, very, very professional, but you cannot see the pointer, right? So you are very professional, but you are not perfect, you know. Don't worry, guys. But you know, when we started studying all this, we saw that light emitting sources will take care of wavelength, of energy output, of, I can use the mouse or what? Okay, thank you. Well, oh yeah, oh no. Well, don't worry, it doesn't matter. So a lot of things were being taken care by the light sources, by the platforms, but you know, many other things were not. For example, the main important thing is the focal distance. People will grab a Petri dish, put some PRP on, some lead array, and just illuminate the sample and say, hey, it's working, it's not working. Yes, of course, because you do not have a treatment. You are not controlling all the variables that are very important. For example, focal distance, scattering, different energy losses, dose homogeneity. You get different amount of energy in different parts of the sample. So of course, this was not a way to go. As a matter of fact, if you know this is the biphasic dose response of light. So if you are here in this part of the curve, you have no effect. If you are here, you have inhibitory effect. It's nothing. You need to be exactly here, on top of the mountain to have effect. So you need to regulate the energy in a very precise way. And this is exactly what we did. We built a machine that is capable of giving light and temperature in your consultation, but controlling absolutely every single aspect of that journey. So basically, instead of having a machine that was illuminating the sample, we had a machine that is capable of dosing the exact amount of energy. So when we want 1 joule per square centimeter on the sample, the machine is not emitting 1 joule per square centimeter, it's emitting whatever it takes so that you get 1 joule in every corner of the sample. And by doing this, we change illumination into a treatment. We really have a treatment, something that is capable of giving light and temperature. So the general idea is to get autologous materials, most of them, not all, we will explain this later, do some processing in the machine, which is touching 2 buttons, and then re-injecting on the patient. We are only using autologous materials, okay? So you could use any sort of plasma, any derivative from the plasma like PRP, ultra-rich, growth factor, whatever. The only thing we are not using is PRF because it will clot inside the kit. I will explain that later. But other than that, you can use anyone. You can use also biopsy derivative products, stromovascular fraction, stem cell concentrates, anything that is liquid and that has cells or platelets inside. I need to biostimulate something alive, a cell or a platelet. So I am not going to put Botox inside the machine. I'm not going to use hyaluronic acid inside the machine. I'm not going to use mesotherapy drugs inside the machine. I'm using liquids that have living components inside. Is this clear? This is very important. We need cells or platelets, okay? So, what happens inside the machine? This is a little bit, technique, but it's important. What happens inside the machine when we use the temperature and the light? Well, different things happen in different protocols. For example, what happens to platelets? This is all very well documented, very well published. For example, many changes take place inside the platelets. For example, phosphorylation of P38 or glycoprotein 2B3A, this will grow up. This is an activation marker. Instead of that, inhibition markers like Vas complex, they will go down. This basically says that the platelet is getting ready to work. We are priming platelets. We are not activating platelets. The platelets are not degranulating, are not sending away the alpha granulocytes. Because if you are doing that, you have the clot. This is what calcium does. So this is one step before. We are priming. We are running the engine faster, okay? You will see this on the microscope. This is an 80-year-old paper because a platelet changes the shape. It changes from an octopus to a spherocyte. This is very well documented. Peripheral tubular halo, reorganization of the organelles. The platelet gets ready to send the granules away. It just builds like the railroads. It's very, very easy to see. It's very interesting. Well, another thing that happens, for example, with temperature, we can use it to increase the release of exosomes. And this is what we use. We use exosomes that are already manufactured by the cells or the platelets. These are called naive exosomes. We are not inventing exosomes. We are not touching exosomes. We are not touching cell machinery. We are not touching genes. Nothing. The exosomes are already there, already created, and we are just getting them out. How? For example, 37 degrees. Basically, when you increase the temperature of a cell, the kinetics inside the cell increase. The collisions between multivesicular bodies, which is the precursor of exosomes, against the inner side of the membrane increase. More collisions means more docking, okay? And then temperature also plays a role in membrane fluidity, so it helps exosomes getting out. And when exosomes get out, you see the third one over there, this is called tethered exosome. These are the exosomes that get attached to the outer side of the membrane once released. These are also reduced by temperature. So basically, by attacking these 3 points, everything happens inside the machine. We don't do anything, okay? But this is the rationale behind the releasing of exosome. This is not only important, temperature. Temperature is an action combined with light, with the calcium coming from blue light. This is what I'm about to explain. So photothermal biomodulation. We use light and temperature to modulate the activity of platelets. This is basically the whole idea. When we talk about light, this is a little bit different than the temperature. Temperature affects all the structure in the same way. Light do not. Light needs something that is called chromophores. Chromophores are structures that change electromagnetic energy from the light to chemical energy. This is what happens in our eyes when we see. This is what happens in photosynthesis in plants. It's exactly the same. So we have a lot of chromophores in all our body, in all our cells, in all our mitochondria. Some of them are very famous, and you know them. In the second year of our medical school, we know phosphorylative oxidation or respiratory chain. This is very simple. It's complex, but you know about that. There are certain macrocomplexes, number 1, number 2, number 4, cytochrome C oxidase. All of them are affected by light. The first 2 cryptochromophores by blue light, the number 4 by red light. Basically, what you will get, you know about all this. I am dashing through this slide. You get a translocation of the protons to the intermembrane space, and when the cell wants to restore it, it does so through a pore in ATP synthetase. ATP synthetase is an helix. It rotates, and basically it generates a lot of ATP. A lot of ATP. So this is another thing we get. Cells fully charged with ATP. ATP for cells, it's like money in our pockets. You can use it for whatever you need to do. Okay? This is very, very important. And this is the most documented effect of light on biological structures. This is very well known. Another thing, a different, for example, chromophores that we have are opsines. For example, number 3 and number 4, these react to blue and green light. Opsines are a different type of chromophore. They are, this is a little bit technical, but basically through a voltage channel, they will increase the amount of calcium, and this will impact on certain processes that we are looking for to enhance them, like, for example, rejuvenation, for example, or processes that are needed. Basically, the idea behind the chromophores is always the same. Any light that acts on a chromophore will release a second messenger. This typically is calcium or oxygen-reactive species or neutral compounds. This will act on nuclear transcriptor factors that, impacting on genes, will affect the proteins needed to wound healing, angiogenesis, fibrosis, etc., etc., etc. This is extremely well documented and known. We just put all this together inside a machine. That's it. Different types of energy, light and temperature, different time exposure, different wavelength, different power in three protocols. That's it. Protocol for PRP, for cell, and for extracting exosomes. And you know, the machine is capable of handling 5 different wavelengths. We are not using these 5 wavelengths. We are using only 3, red, infrared, and blue. Why? Because there is not enough evidence with green and yellow. This is for research and department. Maybe in the future we have different protocols. We are using only 3 temperatures, 4 degrees, 23, and 37. Why? Because there is the evidence. There is not enough evidence to use 7 degrees or 29. The machine is capable of using from 4 to 45. We are not using all of them. You can access the custom menu and you can use any energy any way you want. You are physicians. The industry is not going to tell you how to do things. But there's not enough evidence other than what we use here. 4, 23, 37, red, infrared, and blue light. So basically, this name, MCT plasma, was given by a distributor. We get all kinds of names. They call it super PRP, they call it turbo PRP, they call it Viagra PRP, you know, you name it. But you can get the idea. This is basically the same like a PRP, but super regenerative power. Much more growth factors, much more ATP, much better results, period. So the same indications, the same contraindication. This does not add any contraindication. As a matter of fact, since this is a commodity, we are improving a tool. So now it's being used by dermas, urologists, gynecologists, traumas, you name it, okay? The way they use it obviously differs on the indication. We will talk about this. Because this product is not what we have been seeing during the last decade or 2 decades. People were just, I have a better PRP because I centrifuge double, or I centrifuge 2 times or 3 times. It's just platelet concentration. That variable is exhausted. That's it. You cannot concentrate no more. If you centrifuge a little more, you destroy the platelets. This is what we did when we started. We just were, we were, how do you call it? We're calibrating the centrifuges. That's it. So MCT doesn't do that. We are not a centrifuge. We don't care about platelet concentration. We are just doing something so that platelets performance is better. That's it. So with the MCT plasma, which is, think about it like a last generation PRP, you're getting the same material with much more growth factors. They are being released in a more sustained way. And of course, you have much more ATP. You will see this on results. It is very easy to see. We are having great results. And another study just ran into the fact that it's less painful. I'm not sure about that. We are conducting a study to see if it is less painful and why. Is it because of pH? Is it because of density? Is it because of, you know, subjective feeling of a cold substance? You know, but this is what you get with MCT plasma. Another product, completely different product, is MCT exosomes, and this is what you're really interested in. So we have to go a little bit back and talk a little bit about exosomes, so that then we can see about it. Now everybody's crazy about exosomes. We are changing the production in the journals. We are getting phone calls from the editorials to please do a second edition with exosomes. We are getting invited to different kinds, just exosomes. This is all exosomes. By the way, I will be giving 2 lectures in IMCAS, so if you have time and you are there, please step by. But also, at the same time, with the skyrocketing interest in exosomes, we are getting this. Warnings from the FDA, people injecting things that are not to be injected, you know, all sort of crazy stuff that, you know, regulatory frame, it's very simple. In every country is different. In Europe, it's very simple. You just can inject if it is a medical device. If not, you can use it as a cosmetic, and that's it. That's it. I know here the policy of the government is very good. It's very good. If it is injectable, it is a medical device. That's how it has to be. Because if not, you are not protecting your patients. Because to be a medical device, you have to prove that you are safe and you're effective. If you cannot prove that, then you are not a medical device. This is basic and it's very interesting. So understand an exosome is a way of communicating between cells. But it is not a one-to-one phone call. It's more like an explosion. I explode, and all of you will get my exosomes, all of you. Then inside, you will decide if you use it or if you discard it. The decision on where to use it is inside the cell, not in the uptake. The uptake is non-specific. So we will talk about these crazy things later, okay? Inside the exosome, there are a lot of things, too many things. The most important for us are the growth factors and the microRNAs. But there are a lot of things, absolutely lipids, glucids, whatever, you name it. Very important, microRNAs, not messenger RNAs, not mRNA. mRNA, M-I-R-N-A. This is very important. Once some of you got my exosome, the exosome inside can undergo two paths. Number one, it fuses with lysosome and it gets destroyed and discarded. And the second one is through back fusion, the content will be released to the cell. Okay? So what is the content? The content, very important to us, is RNA, as I told you before. mRNAs, microRNAs, are a very specific type of RNA. These standard RNAs that you know from school, transfer RNAs, messenger RNAs, ribosomal RNAs, are what we call coding RNAs, because out of them, you will get amino acids and a protein. You will code for amino acid to make the structure of your body. This is basically the basic foundation of our body, of our biology. Well, there are other types of RNAs, like microRNAs, that are non-coding. It means you will not be able to get any amino acid or protein after them. So what do? These microRNAs inside the exosomes will get stuck to the messenger RNAs from the recipient cell and will make that thing faster or slower, or they will destroy it. So they have the power to affect these processes. This means that if you need collagen and you are synthesizing more or synthesizing less or whatever, okay? So the action of exosomes, of true exosomes as a regenerative tool, is epigenetic. Not genetic, but epigenetic. It affects an activity with the transcription. And this is very well documented. Very well documented. Though we know much less than what we should and what we will know in the future, a lot of things are already being very well noticed. And you see, this year, 2024, Rubkun and Ambrose, they got the Nobel Prize for discovering microRNAs in 1993. This is the Nobel Prize of Medicine of this year, is microRNA guys that discovered them. And they say that there is a long way to go. We need to understand much more than we do. So the first thing that you need to understand when you face an exosome product is that not all the exosomes are the same. You need to get some knowledge to be able to decide this product is great, this product is not, this product is safe, this product is not. Okay? So you need to understand that some people are working in all these sea of things about the exosome, but to confuse you, not to give you transparency. A lot of people are working for confusing. So it's very different a product that is autologous than a product that is non-autologous. It's very different a product that is injectable than a cosmetic. It's very different a certified legal product than another product that is not. And it's very different a product that is evidence-based than a product that is marketing, as is now surfing the wave of exosomes. They say, all autologous products are saying now, oh, I have exosomes. How do you know? Oh, yes, now I know. You've been on the market for 20 years, and well, now we have, 50 billion. and you close your eyes, you come back, and no, now we have 60 billion, and now we have 70, and it doesn't make any sense. So you need to be very careful about this. You will find all sorts of products on the market. Some of them, we are not going to talk about them right now, which are the illegal products or non-autologous products that are being injected. This is illegal in Europe, this is risky, you shouldn't do it, never. And then you have autologous products that are really great products, like MCT. You have other products, autologous, but I call them marketing products that cannot explain what they do, how they do, etc., etc. And you have non-autologous that are being used topic. You have these mimetic products that don't have real exosomes, but the proteins that are included in exosomes, and products with exosomes. So this is everything you got on the market right now. So we are facing 3 types of problems with exosome product. Hopefully you are not facing this in here, because you are having a strict regulation. From now on, you should be protected, and you have to be smart. Biological safety and efficacy. This is the most important problem that we are facing when we use products that are not certified and safe. But of course, misleading marketing claims and not fulfilling the regulatory frame. We have produced a 60-page document that is free, downloadable, that tackles everything I'm explaining to you right now, but in a very deep way so that you can read it, etc., etc. Please check it out. You will find a lot of good things about exosomes. So, first of all, let's talk a little bit about safety. Number 1, exosomes are capable of an immunogenic reaction. They have proteins like tetraspanines and major histocompatibility complex. They can produce this kind of reaction. They are less antigenic than a cell, of course, but they are still antigenic. So if you work with autologous, you will have no problem. If you don't work with autologous, you might have this problem. And people are using stupid claims like, oh, we are 98% compatible. Okay, we are 99% compatible with an ape. We share the same 99% genes with the chimpanzee. Between you and us, we share 99.9, but if I give you my liver, you will be immunosuppressed for the rest of your life. So 98% is not acceptable. They say, we are 98% compatible. Exactly, I answer, exactly. This is the real problem. Number 2, all of these cross-contamination, they have been published and demonstrated. I'm not saying be frightened, no, this is not what I'm saying here. I'm saying that if you don't have the proper certification, don't risk it. If it's not a legal product, don't use it. If it's not an injectable medical device, don't inject it. It's very simple, very, very simple. Some claims like elders don't release enough amount of exosome. Let's get the exosome from a young rat, because that would be better. Your own exosomes are not good for you. You are 48, you're dead. Well, there are a lot of studies that show that older people even release more exosomes than younger people. And this is very well documented. So, forget about these claims. Just, start working with legal products. Second thing, what about the cargo? It's 1/3 of the cargo of an exosome is unknown. If you are using your own naive exosomes, you are completely safe. If you are using the exosome of a sequoia or a goat or whatever, all of these might be a reality. So let's move forward to what you really want. Effect. If you have a non-autologous exosome, other than producing a immunogenic reaction, it doesn't matter. If it doesn't do any good, it will still be destroyed by your own body. Your own body recognizes that's a foreign thing. So even if it doesn't produce any good reaction, it will make no benefit. So it's enough about this. You understand my point. You need to work with certified and completely safe products. The same happens with bioengineering and naive exosomes. So I'm moving forward, not getting delayed in this. Okay? So what about these crazy claimings of quantity? Everybody is talking about, I have more exosomes, I have more exosomes. Nobody knows the exact amount of exosome that makes the result. So this is like, many times in the past, people fighting for like hydroquinone 2%, hydroquinone 4%, this is a completely different situation. It's very well documented that even in PRP, we can get 70 billion exosomes per ml, naturally. So when people claim, I have 55, I have 60, I have whatever, I have been reluctant to publish this number, MCT, because the truth is framed by the facts, and the people that lie don't have this frame. So you say 149, they say I have 155. It's stupid. It's stupid about claiming the number. But this is only preliminary data. We will publish this during this month. The number is around 200 billion per ml, and with every kit you are using 10 mLs. So we are way above anything that you can imagine. And that is not even the most important thing. The most important thing is this. We have transparency on the source. We have categorized our exosomes. Everybody know what's their size, what's their amount. Some studies show that the standard companies, they won't disclose their sources. We do. We take pictures of them, we publish them. You can see the video going through with the exosomes, growing through the channel that counts them. We don't use this kind of thing that, claiming absolutely everything when we know it's not possible. We don't use this kind of names, smart, intelligent, evil, last generation, 2.0, quantic, specialized. This is all invention. This is all marketing stuff. Forget about that. So let's go directly to what we want right now, which is basically how do we get these exosomes, okay? So as I told you before, we need a liquid product and we need living cells or platelets inside, okay? So once you give that to me, for example, let's pretend I have now PRP in my hands. PRP with the characteristics you want. Any brand, any centrifugation protocol, anything you're doing in your own consultation, you give me that PRP, okay? Next thing, after getting your PRP, I will open one of these kits. This is a medical device. This is a class 2A medical device in Europe. It has 10 mLs. It can allocate 10 ml of PRP. It has here two docks. They are exactly the same for standard syringe or for luer lock syringe. Okay? So you will open both caps, attach the syringe and put the 10 ml in. That's it. You take it out. You close both caps, and you will put it. All this was small videos, so sorry about that, but I'm explaining. So you turn on the machine. At the rear, it has a green button. That's the only button the machine has. You turn it on. There's no, it's easy. You turn it on. After turning it on, you're smiling. I've been working with I'm a physician myself. I was working with physicians and lecturing all the time. So a two-button machine is complicated, so we just wanted one. Okay, I said only one, and we got one. Okay, so you turn it on. On top of that, you will have the USB dock where you will be updating the machine all the time. So it's like a phone. You will never get this machine obsolete so that they sell you a second one. No, just you receive an email, you put it down there and actualize, and you had always the last technology in your machine. After that, you turn it on, you put the PRP on the kit, and then you put the kit on that tray. It's very simple. You just put it here. Directly, it's very simple. This was an animation, but you will not be able to see. And then you put this inside the machine, the tray. Zero resistance, zero difficulty. And it's built in such a way that you cannot miss it. If you put it the other way around, it won't close. If you put it, so it's very simple, very simple. After that, you will see this on the machine, these three buttons. So you have the PRP inside the kit, you have the kit inside the machine, now you have to decide. If you have PRP and you want a super PRP, you just go with PRP. If you have a different solution like stromovascular fraction, you will go with cells. Why the difference? Because platelets are very reactive to cold. We get a huge amount of biological activity when they are down to 4 degrees. It makes no sense doing that with cells. So the program with cells, with no platelets, has no cold. Okay? This is a little bit counterintuitive because many people are heating up the plasma. That is good for solidification, for getting the clot, you know? But for performance of the platelets, 4 degrees. This is great. And the third one is to get exosomes. If you want exosomes, you just touch the button. That's it. Once you have chosen any of these 3, all these things will happen inside the machine. The machine will sequentially use the temperature, the lights, the time exposure, everything will be done by the machine. You are just waiting. The machine will give you the feedback of every session. It will explain to you everything that's happening inside. And once it's done, it will say, I'm done. And so you just take out the tray in the same way. You take out the kit in a very simple way. You open always both caps. They are the same. They are exactly the same. But it's because of vacuum. Since this is a very special volume to surface ratio, the chamber is very thin. So we always have to open both caps. And you extract whatever you've been using. In this case, it's people using stromovascular fraction. You see, it's more hematic. It's not PRP. And after that, you are good to go. You are good to inject it. That's it. 0 problems, 0 complications. It's just one more step in the chain of processing a PRP. Okay? So this process will add 10 more minutes to your chain, but you will get much more power, much better results, and the possibility of regaining all your patients that you already lost to PRP because they are bored. You know, we've been using PRP for a long time. So basically, another thing that is very interesting about this treatment is that we are splitting the dose of PRP into 2. So we get the PRP, we split into 2 syringes, okay? The first syringe, we will use it to get this MCT plasma, which is the super PRP. Once it's done, I will get it out of the kit. I will put the second half. And while I'm injecting the MCT plasma, the first half, the machine is getting the exosomes, okay? When I finish using the MCT plasma, I will use the exosomes. In this way, you will lose less time. It's just the same time as one procedure, and you can use for both treatments the same kit. The kit is not one-use disposable. It's one session disposable. It means if you use it 2 or 3 times in the same session, no problem. The problem is that if you want to keep it there, your patient comes tomorrow and you want to use it again, because the chamber is so thin that you will never be able to wash this problem and re-sterilize it again. But you can use it 2 or 3 times. In this way, people are charging whatever they want for the MCT plasma, which is the last generation super PRP, and then they are charging whatever they want for the exosome. So it's been a very profitable treatment. Of course, we can break this down, but it's not the moment to do this. We can break this down in every single step of the treatment. How are calls and physicians along the world administering this? Well, this is very difficult to say because it depends on the pathology, it depends on the specialist. For example, it is very different to think about 10 ml for phase rejuvenation, 3 sessions, 3 weeks apart from one another, and maintenance sessions during 3 months, for example. That would be a protocol for phase rejuvenation. But a trauma that uses knee osteoarthritis is 1 session, 1.5 ml. That's it. So depending on what's your specialty and the pathology you are treating, it would be one or another. It's AUTOLOGU's completely safe product, so there are people that are more aggressive. They are giving 1 session every week, no worries, and there are people more conservative, 1 session every month. You will see everything. So let's take a quick look about some of the studies and some of the pictures so you can see a little bit. Okay, so MCT, they have a lot of basic, how they build the machine, and that's very interesting. But for us, for example, this is Dr. Hernandez's small series of 7 patients with systemic autoimmune disease. This is very interesting. Great results in patients that she couldn't use with any other type of treatment. For neck sagging. Also, we had this study for neck sagging, Dr. Beltran, 45 patients. You see the journals where it was published. Everything is very, very important, very, very well documented. It's public. It's published. You can get it from the Medline, or you can ask me. I will gladly send it to you. This is the study that ran into the fact that it's less painful. And now we are conducting another study to see if this claim is true. Also, great results. This is Professor Mercuri from Milan. This is a case report of linear morphia. This is a localized sclerodermia. Also, he got fantastic results, but he used more sessions, not 3 sessions. He used 4 sessions, I guess, a little bit. This also changes according to pathology. Professora Tejero in Toledo, the hand rejuvenation, awesome results. My reviews and some stuff you can check on the internet. Growth factor content, hair shaft survival, Professor Naranjo in Madrid, that the hair shaft was able to be transplanted for 2 hours more, which is very good for the transplantation procedure. This is a case report of Dr. Amesti. This is awesome. This is just one case, but this patient is 85-year-old. This is a diabetic patient, and between these two is just 1 session of exosomes, and it's only 96 hours. When she told me this, I was like, Alexandra, is this true? Yes, because then I go to Tanger and I have to put the face on this to other physicians. Is this true? Because for me it's amazing, and yes, it is, and sometimes the results are awesome. For example, Professor Mesana in Rome, he's using it for lichen sclerosus, vulvar lichen sclerosus. Gynecologists are crazy about this treatment. We are having awesome results. This guy here, he's mixing it with fat, with stromovascular fraction. And there are different, he's using milligraft, and Professor Brambiglia is using nanofat, so different types of fat, and really, really getting amazing results. Some studies that are still to be published. This is the one assessing the variable of the clot, of density, etc. And you will also be able to see some other cases like rosacea casing. This is 1 session of exosomes, face rejuvenation. You see this hypochromic lesion. This is a very interesting result. This is hypochromic lesion, this lesion here, due to neuralgia. Exosomes play a major role in melanin synthesis. It's amazing, but they have like this eumelanin effect. If you have a higher melanin process, they will take it down. If you have a lower melanin process, they will take it up. It's a little bit surprising, but it's described, and this is what they do. Of course, AGA, androgenetic alopecia, this is from Valencia, this is from Iraq, this is from Serbia, and you can see the difference. You can tell the difference between an inflammatory scalp and a very healthy scalp. This is very interesting. This is not about hair growth, but it's about health on your scalp. The redness there and the whiteness over there. This is, of course, it's documented with Photo Finder and everything you can dream of. Well, hundreds of pictures of face rejuvenation. Ulcers, also very interesting results in wound healing. Vulvar lichen sclerosus, we are having spectacular results in terms of clinic reduction. People are getting, again, their clothes and playing sports again in 10 days, 15 days, which normally takes 45. In gynecology, they are crazy about this. Some complications about injection on the cheeks with hyaluronic acid. Well, real melanosis, also what we said before about the different type of melanin. Well, all these are cases that, of course, they have different protocols. You need to assess exactly what our calls are doing, but you can be sure about this. We have also very different type of documents that explain all this. We also have great results with psoriasis. You can see it here. You can see it also here. We are very happy and excited about this. So check this, and I will be just ready to answer your questions. We produce lots of documents, of course, commercial document explaining on the product. This is standard of any company. But we are also producing this kind of technical documents that, for me, this is a pride in doing this because it's a scientific department. Scientific Research Dossier, this is the 2nd edition. We are explaining all the studies that we have published. So far, we are around 16, 17 studies. Top questions, 50 questions asked that we get all the time. Very short, 3 lines, how we answer all these tough questions. tackling the exosome problem. This is a 60 pages dossier that talks everything you need to know about exosomes in the world of exosomes, not only our product. This is transparency for you. And of course, before and after cases with the explanation of the protocols and all you need to know. But you see also we have this. These are certifications worldwide. You know, you can see our country over there, second top, Morocco, but you can see also European MDR. MDR is a very strict regulation. We are the 9th company in Spain to get it. It's amazing. So, you know, that is because we were able to prove that we are safe and effective. If you cannot prove that, they will not give it to you. They don't give it because you are nice or beautiful or awful. They just don't give it to you. So all the cosmetic products don't have this because they cannot prove they are safe and effective. It's very expensive, it's very effort consuming. So, you know, this is a very important and very safe slide for you. And also Canada, Brazil, very tough regulation. And this is the last one I like to show it to you all the time. We build tough. This was a machine that fall from here. It's destroyed, but it's still working, you know. So, guys, I want to thank you very much for staying at the end of this. And if you want, we can talk and I can answer questions if you are not so tired. So thank you very much.